Hi everyone, my name is Chen Li. Thank you for the invitation to present our latest research in metabolomics and metagenomics in the TEDdy study. Um, so today we will present some research results about the plasma metabolome and gut microbiome in an early childhood study of type 1 diabetes. First of all, I would like to give some introduction about the TEDdy study, the environmental determinants of diabetes in the young. So the TEDdy study enrolled children by screening for their HLA-DR-DQ haplogenotypes from 6 clinical sites in 4 countries. And 3 clinical sites are in US, they're in Colorado, Washington, and Georgia. Um, and another 3 are from Sweden, Finland, and Germany. So there are about 8,600 children enrolled in TEDdy, and they all have higher genetic risk of type 1 diabetes compared to the general population. And after the enrollment, the TEDdy children were followed up every 3 months after from birth until they're 4 years of age, and then they switched to a 6 month follow-up schedule after 4 years of age. There are 2 primary endpoints of interest in the TEDdy study. The first primary endpoint is islet autoimmunity, which is symptomatic, and it is diagnosed by the first persistently confirmed autoantibody, including GATA, IA2A, or IAA. And the second primary endpoint in this study is the clinical onset or clinical diagnosis of type 1 diabetes based on the autoantibody and HbA1c level. So the TEDdy study aimed to identify the environmental triggers or exposures that are associated with the development of this disease, either IA or T1D. So the consortium collected different types of data, such as the exposure data in diet records or their physical activity, or the vaccination records and infectious disease episodes. There are also some other data types that the TEDdy study collected from their either stool samples and blood samples, for example, the genomics and multi-omics data. So the stool samples of the TEDdy children were collected every month by their parents and then shipped to the repository assigned by NIDDK. Uh, and then the blood samples of these children were collected at their scheduled clinic visits every 3 months. And so the stool samples will be used for the profiling of, for example, gut microbiome or virome, depending on the reference database they use. And the blood samples can be used to measure nutrient biomarkers, for example, vitamin C, vitamin D, and the fatty acids, or the high dimensional omics data like metabolomics and metabolites abundance, or gene expression, or protein peptide abundance. So these data sets were all collected for the children in TEDdy. Although I cannot present or cover all of these data types, but it's a very large cohort with different types of data in addition to their exome-wide genotypes. So in order to provide multi-omic data, it's difficult to measure that for every participant in the consortium. So the investigators use a nested case control design to select a subset or a subcohort of these children to perform the omics data profiling. The nested case control design is a little bit different from the case cohort design because the controls that they're going to select are determined by the age of the case's diagnosis. For example, by the end of the design date, if there is a subject or a child developed the disease of interest, say IA, at their age of 0.8-year-old, then the investigators will randomly select two other participants from those who have not developed the disease by the age of 0.8-year-old. So, and then these children will be matched, and then we call them either risk set or stratum. And these children are also matched by the other factors like clinical site, gender, and family history of T1D. So under this design, the best approach or the robust approach to analyze predictive biomarkers would be conditional logistic regression. In this method, the comparison is performed within each match stratum or match risk set. So we compare the case to his or her match control subjects instead of comparing all the cases to all the controls simultaneously. And also within under this design, it is difficult to analyze the time-dependent risk because their status of the disease status were all fixed at a matched endpoint. And the profiling of the omics data under this design are all applied to the samples before the matched endpoint for each risk set. Okay, so how does the omics samples look like in terms of the age? So this is the distribution for the stool samples. It is from a publication based on the microbiome data. So as I mentioned, each case, for example, for IA case, and the samples, the stool samples were collected and provided for microbiome from birth until they are diagnosed as IA or seroconversion. And the match controls will have the samples profiled at the same time point or age point up to the ending point matched to the case. So if we visualize each subject samples using a line of points, then you will see for the match controls, I would prefer not to call them healthy controls. I would call them just controls, NCC controls. Those control subjects have their samples truncated by the age of their matched cases endpoint. So if you look at the cases for IA or T1D, they show a similar curve for the ending sample, and you'll see the same for the controls that's selected to match them. But those control subjects are not always free of disease. They might develop the disease of interest for either IA or T1D at later age. So that means for these subjects in this left panel, they might they will have some missing samples between this ending point and their actual disease onset age. And in TEDI, the plasma samples were also used to provide the metabolomics data, and the distribution of the plasma samples would be similar to the stool samples. And the metabolomics data were provided using the GC and LC mass spec platforms, and two types of metabolites were measured. The first type is the primary metabolites, for example, the sugar alcohols or the amino acids. And the primary metabolites usually represent the growth or development of organism. And the second type of metabolites provided is the lipid species, or sometimes we call that lipidome. And the lipidome include the fats, fatty acids, or its derivatives. And it is kind of like a marker for the nutrient intakes or energy intake, as well as the signals for gene regulations. So in the metabolomics data, we have about almost 400 or 500 metabolites, but we also have a few hundred unknown metabolites. Those are unannotated metabolites. And the metabolomics data processing follows the standard bioinformatics pipeline, starting from the peak detection and then quantify by the area under the peak. And the missing peaks was first recovered by backfilling and extracting the missing peaks. But still, at the end, we have a small proportion of missing peaks or missing metabolites. And then we apply a machine learning method to impute the abundance table for all the metabolomic samples before we perform any downstream analysis. And in our research, we first review the previous findings in the TEDI study. So when we reviewed all the previous findings, we found there are two major types of the first appearing autoantibody that define the IA, that is either IAA first or GETA first. And these two types of autoantibodies show different incidence curve or incidence pattern in either all the TEDI participants or the general population without a family history of T1D. So for the IAA first, we can see there is an incidence peak around later age of infancy, and then it dropped sharply. And but for GATA first, we can see that during the age infant age and the age between 1-year-old and 2-year-old, the incidence rate increases sharply, and then the GATA first incidence will stay at a relatively stable level. So we were interesting about the each type of IA, depending on whether we want to find out the biomarkers for the risk of IA first, GATA first, or whether we want to differentiate these two types of autoantibody. And we also saw that in the previous analysis and research that the earlier onset of IA would signal a faster progression rate to T1D. And these results are all based on a full cohort analysis. That means it's based on all the available, like 8,000 children enrolled in TEDdy. And previous research also found some biomarkers under the NCC subcohort design. For example, the vitamin D will reduce the risk of IA for either type of autoantibody, and vitamin C reduced the risk of IAA first, but it did not reduce the risk of GATA first. And we used the primary metabolites to perform some similar analysis using the conditional logistic regression. And we found IAA first and GATA first were signaled by multiple metabolites at infant age. Some are similar, some are the same, but some are distinct. So in order to answer the question that whether there's difference between IAA or GATA, or if there is any biomarker that differentiate the early incidence of each type of antibody, a key question is how do we define that early incidence for each type of autoantibody? So the question will become to find the subgroups within the cases of IAA or GATA. And it's hard to justify that infant age is the best cutoff age, given the incidence curve of GATA, for example, like here, it's still increasing after infant age. So we don't want to manually cut off that age, and that's why we apply the unsupervised clustering method to identify any possible grouping among these cases. So we selected about 165 children having their lipidome profiles available at 3 consecutive visits. Those visits are the IA diagonal, the seroconversion visit, and the 3 months before that, and then the 6 months prior to that. So it's during the 6-month window. And we merged the lipidome profiles for each of these children so that the dimension for these features is the original dimension of the lipidome times 3. And we used the Gaussian mixture model to perform this unsupervised clustering because the strong correlation or the network structure between lipid species, similar to the gene expression co-expression network. So we use this Gaussian mixture model to identify 4 clusters or 4 groups of children. And we also compare, based on the previous findings, we also compare the vitamin C and vitamin D level of each group of these children and see if there is any difference between them, since lipidome is closely related to the nutrient markers. When we visualize these clusters, we saw using the TSNE plot, we saw the distance between these 4 clusters. And when we look at their age of IA seroconversion, that is the onset of IA, then we saw these clusters are not just simply the age group. At first, we thought that maybe the unsupervised clustering would just group these children by the age of their samples, their lipidome samples. But when we look into these groups, these clusters, by the type of autoantibody, it looks a little bit different from what we expected. For example, for cluster 3, we do have earlier age for this cluster, but we found that for the IAA subgroup, in this cluster, it includes almost all those early incidents by the end of infant age. And also for the GETA first, this cluster included most of the early instances of GETA. And we still have the subjects who has plasma samples at a similar age in another cluster, but they are not grouped together. So that is an interesting finding that we actually not just finding the age group using this unsupervised clustering approach. We actually kind of latently detected the early and the late onset of IAA or GETA. And we also look into the progression risk from IA to T1D between these clusters. And then we found another cluster, the cluster 4, the yellow, the color by yellow. They progress to T1D at a lower rate. So now we are more confident to cut off the cases by their incidence age. For IA, we use a 15-month, and for GETA, we use a 24-month. And then we perform differential analysis to detect any biomarkers like in the metabolome, including lipidome, are associated with the early and late incidence, and as well as the T1D progressor and non-progressor. So this is the results in that differential analysis. So for the early IAA versus late IAA, we saw the cholesterol metabolite and the vitamin C are associated with early onset of IAA. But all the other lipid species that we identify are associated with early onset of GETA versus late onset of GETA. And among the T1D progressor and non-progressor analysis, we just found multiple lipid species that differentiated progressors in GETA versus the non-progressors in GETA. We didn't see any significant markers for the progressors in IAA. So these are the previous results that we have found, and it has been published in Diabetes. And given these results, we are wondering whether this age effect can be seen on the microbiome. So there is a publication based on the microbiome, and they also use unsupervised clustering, but they didn't group the children. They grouped all the samples of these children. So they didn't merge their longitudinal samples. In the other way, they actually just grouped the sequencing counts using dirichlet multinomial clustering to group all the available samples of the teddy children. And the results show that these children's sample or the gut microbiome can be defined into different phases. And the earliest phase is overlapping with the infant age. So it looks like that there is age effect on their microbiome sample. And in this study, we believe if we subset the subjects to be those who have earlier incidence of IA and also use their early age or infant age microbiome sample, there might be some microbial signals that we can identify. So this is the general hypothesis after reviewing all the previous results. The teddy microbiome data there are two platforms being applied in the microbiome data, either 16S or the metagenomic, the whole genome shotgun sequencing. But in our research, in our recent analysis, we just focus on the whole genome shotgun sequencing data. It starts from the standard procedures like DNA extraction, library preparation, and then sequence on the HiSeq 2000 platform. And it was aligned using their in-house pipeline. And the abundance table was generated at operational taxonomy unit, the OTU level. And the annotation in the teddy data is at a species level, although we can aggregate it to a higher rank level, like genus level, family level. And the abundance table, usually what we did is to analyze the composition data for microbiome. And some researchers probably want to look at the presence of each taxon, but we haven't looked into that part. We probably just recode it as 0 or 1 if the abundance is 0. When we visualize the temporal gut microbiome data, There is some pattern in some of the taxa, but it's hard to tell the strong impact or the strong effect of IA. Why is that? We expected to see some change in the control group and or the case group over time, but we didn't see any strong difference. I think one of the reasons is the exposures in TEDdy were not were different, kind of different from the experiments or clinical trials, because this is an observational study. So all the exposures can occur between 4 months of age and 9 months of age. And that means we probably, for example, a child may experience the change of their dietary intake, like a stop of breastfeeding at 6 months of age, and they may also experience infectious disease episode, or they have vaccination. And these time points are not, there is no intervention of these time points. And that's why the microbiome data in TEDdy study are kind of we have to say noisy, but it's not because of the technology. It's probably because of the design, that it's an observational study and it is the infant age, and the children have a sudden change of their dietary intake, breastfeeding, and infectious disease. So it's hard to tell whether there is a signal. And among the children that we selected, most of them are IAA first or early GETA first. So now we have, by looking at existing data sets we know their HLA haplogenotypes, and we know that DR3, DR4 is a strong genetic risk for either IA or T1D. And then we identify a few metabolites at every 3 months at infant age. And now we have candidate microbes that we assume associated with IA. So if we want to build a model to predict IA or to analyze their association with IA, the traditional thought would be using the conditional logistic regression per time point. And we did, but when we did that and correcting for multiple hypothesis testing, we got some inconsistent results at different time points. For example, we identify some species, for example, at this time point, 3 months or 4 months, and then later we cannot see that, or even we can see a different direction of that effect. So it's hard to draw a conclusion by just looking at all of them by every time point and then correct for multiple hypothesis testing. So we decided to use a longitudinal model, that is the linear mixed effect model, to analyze or to compare the microbiota between cases and control. I think there has been a lot of tools in the microbiome study using or implementing the linear mixed effect model. Some suggest a transformation of the composition by log abundance or by the arcsine square root. In our current research, we try to avoid a discussion or the selection of transformation. So we use the zero inflated beta linear mixed effect model, which accounts for both the composition and the presence at each time point. So this model assumes the composition, relative abundance of each taxa follow the beta distribution, but the probability, and it also assign a probability at a zero abundance. And there are two submodels in this method. That means we can use different random term to account for the correlation between the samples for each subject. And joining these two submodels, if this is an experiment or clinical trial, then the group variable would be the treatment, or sometimes we can use, for example, phenotype as the group variable. So that's the common way that we apply this model to longitudinal microbiome data. However, in TEDdy, the group variable that we can use is a disease outcome, that is IA. So at first we thought it's fine then using IA as a group variable, just like treatment. And then we can use a time-invariant or time-variant covariates to fit the model. And we also allow the subject-level random deviation using this approach. But once we try to include the covariates, we had some questions about which covariates should be excluded or included, because we know that IA, this group variable, is confounding with multiple either exposures or demographic information, like clinical sites. So that's the first question. And the second question is still multiple hypothesis testing. Once we correct for correct p-value, then we probably need to use a q-value cutoff as high as 0.3 sometimes in order to identify some microbiome features. So we came back to think, is it because of the group variable that we use is not really uh, the same as a treatment or phenotype? So we decided to revise this model by assigning a randomness to the disease outcome. So suppose disease outcome is O. And then for each subject, the outcome can be modeled by a logistic regression, just similar to our previous analysis in TEDdy. But here, I simplified the model to be a regular logistic regression. We do have an advanced version for this model, but I'm not presenting here because the limit of time. So suppose we have just regular logistic regression for the disease outcome, and then the known risk factors can be included in this logistic regression for disease. For example, HLA haplogenotypes and the metabolites that we identify at a certain time point. And similar to the previous method, the ZRBR method, we use a random term, epsilon, to account for a subject level deviation in this disease risk. And it could be, this deviation can be caused by any possible factors or exposures. But we do not need to specify those exposures in this model because there are too many and we don't know when that occurs sometimes. And for the composition or presence of the taxa, we use the similar framework. So that is using the logic link function, and then we allowed the time variant variable, which is the age, and we use the same random term, the random effect from the logistic regression model to account for this deviation in the risk, because we assume that this latent deviation in the risk is associated with the microbiome composition. So this is the model that we built, and our final goal is to answer the question whether this gamma coefficient is 0 or not. If this is 0, that means there is no association between the microbiome composition or presence and the disease outcome or the disease risk. So we join these three submodels by join the likelihood, and then we estimate the parameters by a shrinkage approach. And then we use the wall test to answer the question whether there is association or not. Okay, in order to illustrate the performance of this model, we need to design a simulation study which needs to mimic the sequencing counts instead of the composition data. Why do we need the sequencing counts? If we just use a beta distribution to simulate the composition data, then it could be any type of omic data. It can be fatty acids as well. So in order to make sure this is the data sets similar to the metagenomic sequencing, we use a Dirichlet multinomial distribution to mimic the counts. So in this metagenomic sequencing, it starts from the true composition vector. For example, in this sample, the composition or the relative abundance for those bacteria can be assumed as like 1/3. This is just a simplified example. And then after the DNA or RNA extraction and the PCR amplification sequencing, then what we observe might be a little bit different from the true composition value. So this is the idea of the simulation of sequencing counts in metagenomic studies. So we probably need a baseline parameter value from the real data set, which is estimated from the TEDdy data. And then we are going to simulate the true composition vector for each sample and at each time point. But when we try to shift the baseline parameter to by the age and by the group, the case or control, we had a little challenge in this shifting. This challenge is caused by the restrict of the composition vector, because this vector sum up to 1. For example, if we shift taxon 3's composition from 20% to 42%, then that means you also need to change some other taxon in the same sample by reducing their composition level. So the data generation for metagenomic counts or this composition is a little bit different from the RNA-seq read counts, because in the RNA-seq gene expression counts that we can just shift for some genes and then the other genes can remain the same. But for metagenomic simulation, we need to consider the shifting for the entire at a community level. So that's the biggest challenge. And what we did is to increase the percentage or the composition level for a certain percent of taxa. And then we reduce the same amount of that shift for another percentage of the taxa. And then we generate a true composition vector for each sample. And we use the duration data generation function to generate a vector for each sample. And the library size is sampled from the Poisson distribution. And then at the end, we use this composition vector and the library size to generate the sequencing counts for each sample. And then that will make the total OTU counts table. Okay, so we simulated the counts and then converted the counts to the relative abundance for each sample. And the taxa mean relative abundance, suppose we have 1,000 taxa in the simulation design. And most of the taxa had a relative abundance below 1%. The mean relative abundance across sample is below 1%. And we still have a few like abundant taxa in the simulation, but it's fine because we observe similar number of taxa in the real data. And then we evaluated the type 1 error by using alpha level at 0.05 and 0.02, and also vary the filter value from about 0.1% to 0.001%. And it looks like when we raise the threshold of filtering, then there might be more positive, false positive, but it doesn't mean that it's not good. So sometimes we may need to we still need to filter the taxa, but it just depends on which is the targeted taxa or the species that we want to analyze. And this is the simulation for a scenario with both associated taxa and non-associated taxa. And then we analyze the coefficient for their presence association with disease outcome, and another parameter, which is for the association between composition and the disease outcome. After applying this filtering threshold, the performance looks pretty good, but still, when we lower that threshold, we may not have exactly the same performance for each coefficient. And at the end, we apply this method to the TEDdy metagenomic data. We use the samples between 4 and 9 months of age, and the subjects that we selected are the IA cases having the endpoint status before 24 months of age and their match controls. So we also use the non-IA associated factors like HLA, DR3, DR4, and also a metabolite that we identify. We have already identified associated with IA risk at the end of microbiota trajectory. And from 14 commonly seen genera, we selected about 128 species by applying the relative abundance filter and the presence filter. Um, After using these three submodels jointly, then we found 20 taxa associated with IA longitudinally. In the presence analysis, I believe there might be some false positives, but still we have a lot of taxa associated with IA. So in this method, we did find the taxa that cannot be found using the existing methods. And in this method, we also reconfirm that HLA-DR3, DR4 is associated with IA using the first submodel, that is the logistic regression. And we also confirm among these 41 taxa analysis, and the metabolite that we selected is still associated with IA. But why there are only 41 taxa showing this association for the metabolites? I think there are two possible reasons. One is we use just a sub data set, a subset of the children. And another possible reason is there might be interaction between the metabolites and some microbiota species. We don't know when or which one is showing the interaction, but that can be a future topic in our research. And this is the examples of the detected taxa trajectories. I cannot show the name of taxon because it's overlapping with some of the ongoing analysis in the TEDdy study. But we can see there is an obvious difference between their relative abundance at a log scale, although that difference is not always the same across different time points, but we can detect it by using these three submodels. So our future work actually can include a lot of topics based on these three submodels. For example, we haven't included the NCC match sets in the model that I just represented, but we're working on that. And we can also select the metabolites from the entire metabolome by using a feature selection algorithm. And like what I mentioned at the end, the interaction between metabolites and microbes is a key factor that might cause the smaller number of metabolites being associated with IA in the multiple hypothesis testing. And we're also wondering whether we can develop a model for the community level analysis in the future. So these are all the topics that we would like to go further based on what we have developed. And I would like to acknowledge my co-authors, Kendra and Dr. Christopher and Dr. Hu, and they provide valuable advice in selecting the data sets and also interpreting the data, and, as well as the simulation study. I also want to thank the NIDDK Information Network dkNET New Investigator Pilot Program in Bioinformatics, which is funded by NIDDK. And still, I need to acknowledge all the help and the discussion in the TEDdy study group, all the colleagues at Data Coordinating Center for helping me profiling data and also running the simulation study. And I also want to thank all the funding agencies that support the TEDdy study. Right. Thank you very much.
Hi, Dr. Li. Nice talk. So I have one quick question about the IA status you are choosing. So you're using like only up to like 4 years old, like the IA information, and then to do the IA prediction. So I just wondering why like not the later age. I think you're muted.
Sorry. You mean the endpoint age of the IA?
Yeah.
Is that
yeah,
okay?
Yeah.
We used the IA endpoint age before in the microbiome study. We used about like before 24 months of age because we found in the lipidome analysis we found the earlier onset of GATA is different from the later onset of GATA first. So that's why we cut off at 24 months of age. And for IAA first, it's hard to separate them given what we found. We can say the earlier IAA is quite different from the later IAA, but we used the time points. We used the IAA endpoints at 12, 15, 18, and 24, 21 and 21 months of age, so that we have enough sample size. And to be honest, in order to run an analysis, we need enough number of subjects. And the predictive markers, the microbiome markers, are profiled at age between 3 months and 9 months. That actually requires us to select the subjects after, having disease after 9 months. And then given what we have found in the lipidome study, so we restrict the subjects to be having endpoint status, endpoint events between the 9 months and 21 months.
Okay, I see.
Yeah, but we can have more. But it's just like if we have included later, get at first onset at later age, we don't know whether that results will be better or whether that results will be mixed, because they might have different like etiology or different omics signal. So far, we just look at a subgroup of children.
Okay, thank you. Yeah.